All right. Last time we started the functional areas of the brain, organizing the content on the 5 vesicle brain, and we got through the first 3 at the end of Wednesday's lecture. Are we all on the same page?
Yep.
Okay, I put the abbreviated outline. I mean, I took up 2 sections here last Wednesday, and I'll put it over there. So this is abbreviated. A lot of stuff missing. But I'm going to try to hopefully, as I finish up metacephalon and myelencephalon, hopefully tie it to things we've already talked about before. For example, the pre-central gyrus is the primary motor cortex. This is what initiates skeletal muscle contraction. But we have various things that are involved in the motor circuit, and if I put a red asterisk, that's all motor circuit. Okay? So it may be the same colors, it may not be the same colors from Wednesday. Just kind of go with it, right? Because I'm winging it. All right, so now we're in the hindbrain. The hindbrain in the 3 vesicle brain was the rhombencephalon. It splits into 2, metencephalon and myelencephalon. But both of these are considered hindbrain. Metencephalon is the upper hindbrain, and we basically just have 2 structures, and then also the majority of the 4th ventricle. This is the 2nd largest of the 5 vesicles. So we have the pons. This is on the anterior side of the metencephalon. The posterior fibers are continuations of these axons that will go through the cerebral peduncles. So where it says deeper fibers travel vertically, ascending and descending, those are the deeper fibers. Now, when I was a student, our outline had a lot more content than you guys have. So I would do something like this. If I already knew that green is going to be the ascending and descending tracts, and I tied it to cerebral peduncles, all I'm going to do is write deeper fibers in green. Those are the same axons. Same axons. They're just continuing. The anterior fibers are going horizontal to the cerebellum. Since they're both metencephalon, that's why they're horizontal. Okay? That's what it says. Surface fibers are horizontal going to the cerebellum. We got cranial nerve nuclei. And what's nice about this in the brainstem is it's in consecutive numbers. So you just got to remember it's 5 through 8. It doesn't skip around on you. It's just 5, 6, 7, 8. You guys remember on old Olympus towering tops? And then we have this, and this is one of the reasons why organizing the content like I'm doing is really crucial when you get to content like this. Because a lot of this sounds similar. So let me use another color. I will use orange. This is the RCC. Respiratory Control Centers. And there's 2 of them. Yep, new stick in the pneumotaxic center. Now, when we get to the last of the 5 vesicles, myelencephalon, in the medulla oblongata, there is this thing called the vital reflex centers. There are 3 sets of vital reflex centers, and that's going to be crucial for Lab 11.
Okay.
And in the vital reflex centers, one of the 3 is the RRC. And if students are not actively engaged, they're going to confuse it and get the Scantron question wrong. You have to keep it straight. This is the RCC in the pons. And they work together with the RRC in the medulla. In this particular case, the best analogy for the RCC is like that person who likes to drive with both feet even though they don't have a clutch. Have you guys ever seen somebody put 1 foot on the gas and 1 foot on the brake and they just never move their feet?
No.
It drives me nuts. Just let me off a walk. That's how it is.
That would
The abductic center is constantly got the foot on the gas. And the pneumotaxic center, if you read the words, it's right in there. The pneumotaxic center pumps the brakes. And you find a balance between under inhalation or respiration and over ventilation. You're trying to find a balance, and you've got to fine tune things, but you have to work together with the RRC in the medulla. The pneumotaxic center, I think your textbook refers to the pontine respiratory group in the pons. But that's the pneumotaxic center. Okay? Everybody got this? All I'm doing on the whiteboard is showing you how to organize the content so that you can know it. As students now moving closer to point B, you started in point A in anatomy. Point B means you accept the fact that you don't got a choice. It's not optional. You got to know it. Now, you got to come up with strategies so that you can know it. But it's not an option to know. Okay? There's the pontine respiratory group, pneumotaxic center there in green. In the pons. The other big structure, this is the second largest structure in the brain, in the metencephalon, is cerebellum. It has been known for a long time as the proprioception center. Can someone tell me what proprioception is?
Balance, equilibrium.
Yeah.
What's that?
Equilibrium, balance.
Somewhat. You could be more precise.
Like where you are in space.
It is your brain getting sensory information about where your body parts are in space. Where your body parts are in space without having to see it. You can actually feel that your hand is behind your back without being able to see it. You need proprioception to get your wallet out of your back pocket. Everybody got that? Even where your jaw is in space. The most fine-tuned proprioceptors are in the periodontal ligaments of your teeth. There are really coarse ones in your TMJs, but those are really coarse. The fine-tuned proprioception of where your jaw is in space is in the periodontal ligaments around your teeth. Okay, now this is again another illustration of point A and point B. When you understand that this stuff, you don't have a choice. You got to know it. Then when someone comes to you and unfortunately, I've seen too many people like this, and still see them today on social media. There are people who are born ignorant and gonna die ignorant, and there is nothing you're gonna say that's gonna change that. In fact, you can tell there's nothing anybody can say that's gonna change that. And that is a sad thing. You gotta be determined not to be one of them. But now try to explain to someone like that why, when they come in, Doc, just pull out my last tooth. I don't wanna deal with dental bills anymore. I just want dentures. Are you gonna explain that the compression of dentures on the alveolar process causes it to resorb that much faster? You know that because you weren't given a choice. You had to know it. And I try to explain it to someone who is not interested in reality. Or, you pull that last tooth, you are gonna lose center bite. You might think you know where center bite is. You've lost fine proprioception on the location of your mandible. How do I know it? Because I wasn't given a choice. Now I gotta explain it to someone who doesn't wanna know. But that's proprioception. And you need sensory input to be coordinated with skeletal muscle contraction. So if you want to be able to have coordinated movement, you have to have sensory feedback, and that sensory feedback is proprioception. That's where it's happening. And it's going to work together with the red nucleus, basal ganglia, and the motor cortex. Do you see why you want to be keeping all these sections straight? Boom, that was covered Wednesday. Boom, boom, Wednesday. That is just showing you all these things that are part of this motor circuit. It's all in red. Do you see that all listed here? This is the difference between how you all moved now versus how you all moved when you were 2 months old. Hey, open your eyes aren't even pointing in the same direction. When you get feedback and these motor circuit synaptic patterns get fine-tuned, now you move like the way you move. Everybody got it?
Yeah.
As the second largest structure in the brain, this bottom box is not surprising. Even though it is metencephalon, there are some higher functions in this large structure. Certain non-declarative memories. Oh yeah, that's your.
I don't understand what
Let me do this. We were talking about this, we were talking about language. Learning and memory. And then when we're talking about learning and memory, we talked about declarative memory that you guys need for lecture exams, non-declarative memory like swimming and riding a bike. A lot of those non-declarative memories are stored in the cerebellum, especially because of complexities of certain movements. But surprisingly, there are certain forms of schizophrenia and autism for where the pathology is all the way down here in the metencephalon. So that is surprising because it is hindbrain. It is not surprising because it's the second largest structure in the brain. Everybody got that? All right, now we're finally to the myelencephalon. mesencephalon, and that's the medulla oblongata. So everything listed here are all parts of the medulla oblongata, like the pyramids and olives. Those are anatomical structures. The anterior median fissure of the spinal cord continues right at the brainstem, all the way up through the mesencephalon. On either side of the anterior median fissure, here's the number 3, are the pyramids. And you look at that and you think, well, that doesn't look like a pyramid. Well, if you actually cut it in cross-section, it looks like this. The number 3 would be right where my 2 thumbs are touching. The number 3 is the base of this triangular structure. That's why they call them pyramids. Then if you look at olives, if you take an olive, slice it in half, and stick a half olive on one side and stick a half olive on the other side, that's number 2. Everybody got it? And again, this is continuous with the spinal cord. Ascending and descending tracts. Also more on the posterior. Even though the green is listed 3 times, these are the same continuous axons. They're just running through the brainstem, through the posterior fiber tracts of the medulla oblongata, the deeper fiber tracts of the pons, and cerebral peduncles. Same axons. Pyramids is where we see decussation.
Decussation.
Not 100% of decussation, but a lot of decussation is happening at the pyramids, and that's where things are crossing from left to right, both motor and sensory. If you want to contract muscles on the left side of your body, it has to start on the right precentral gyrus. If you are touched on the right side of your body, it's going to go to the left postcentral gyrus. Everything crosses left and right. That crossing left to right is called decussation. And it has cranial nerves, nuclei. And it's 7 through 12. Again, it's consecutive. So both in the pons and in the medulla oblongata, the cranial nerve nuclei is consecutive. 5 to 8, 7 through 12, and that is telling you the nuclei for cranial nerves 7 and 8 are big and are in both. 7 and 8 are in both pons and medulla. Okay, this is key. Vital reflex centers, that's the VRC. There are 3 of them. A, B, C. Here's the RRC. This is a respiratory control center. two of them. This is the Respiratory Rhythmicity Center. And if you are not engaged, you can mix these up, because they're so similar. RCC and RRC. Capitalized word is the difference between RCC and RRC. And they work together. And the orange one. But they are not the same thing. One is in the metencephalon, one is in the myelencephalon. Then you have the CCC and the VMC. This is controlling both heart rate and strength of contraction of your heart. This is controlling regional blood vessel diameter. Not the real large elastic arteries and veins. Not the real tiny one, but the medium-sized arteries and veins. But those are the three regions of the vital reflex centers. Next to this slide, you should write Lab 11. Okay. There are actually other ones, but this is what you need to know for lecture 10-2. Things like your vomiting center is also in this as well. Things that are all pretty crucial. Okay, we're going to be talking about some integrated systems. And the base of one is found in the medulla oblongata, the base of the reticular formation. So, I don't know. Read this. So here's the 5 vesicles. Reticular formation is a collection of gray matter that actually extends 4 out of the 5 lobes. It starts down in the medulla, but as it says here in the spinal box, it goes all the way up to diencephalon, 4 out of the 5. Everybody see that?
Mm-hmm.
Okay. So this I got from the internet. I thought, yeah, this is almost like exactly everything that I cover. There's the anatomy, the physiology, it all matches up. Okay? Go with my words though. But just know that I'm not pulling all these words out of my ass. This is actually well-known brain functional areas. Okay? Is this helpful at all? Connecting some dots, putting your brain's meat hooks into the content, that's active learning. And you need to do that with material you do not have a choice about. All right, so here's the integrated systems. First one is the limbic system. We're talking about this when we're talking about learning and memory. This is very important with the limbic system. And the limbic system is really diencephalon, telencephalon. And again, the gray matter forms what looks like to be rings. And the word limbus means ring. And these are the various parts of it. You can see that thalamus and hypothalamus contribute to the limbic system. So the little pink asterisk, boom, 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 is limbic system. When we were covering learning and memory, we were really talking about the hippocampus, and that's one of the somewhat current areas of neurological advancement is hippocampal neurogenesis, things that I'm telling you about. Amygdala is sometimes referred to as the gateway to the limbic system, not 100%, but a lot of the things that enter into the limbic system enter through the amygdala. That's why it's called that, but that's not 100% true. And you can see here it looks like rings, like 3 rings. Okay. So it is mostly D, mostly diencephalon, but some of it goes to the medial telencephalon as well. Go ahead and fill in the blanks here. Okay. In textbooks, you will often hear the term primitive brain. When people refer to the limbic system, they refer to it as the primitive brain, as opposed to the prefrontal cortex, an area of higher intellectual processing, ethics, planning. This is base emotions, base decision making. You want to avoid punishment. You're searching for pleasure. I'm hungry, I'm thirsty, I'm horny. This type of base level of thinking comes from the limbic system. That's what's in the top box. Everybody got it? Now the Papez circuit is an explanation of a neurological explanation for emotional behavior, especially as it relates to these base emotions. And you can see here that it works in a circle. Cingulate cortex, hippocampus, hypothalamus, thalamus, cingulate cortex. Boom, 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 boom. This is an explanation of why some people can't break out of it. You might know someone like that. You might be someone like that. Man, when you get pissed off, you just can't snap out of it. When you get depressed, you just can't snap out of it. Does this make sense? Mm-hmm. You are supposed to be able to get input from the upper centers of the brain that should kick you out of this thing, and some people just don't have a really good connection with that. And they just stay in this, you can just see their emotions just going down, swirling down in a toilet. Can't break out of it. That's a Pappas circuit. Now, look at that, the 4 things of the Pappas circuit, and tell me, are you gonna do well in lecture exam 2 if you are emotionally upset? It occupies the hippocampus. Where is long-term memory stored? Obviously, you need to work on your long-term memory. Hippocampus. If you are emotionally upset, don't be surprised if you can't remember shit. I knew it yesterday before I was upset. It's gone, baby. And this is why in the Strategies for Success online tutorial, why it says in the first couple of sections on support and preparation, you cannot do these careers. You cannot do these classes in preparation for these careers unless you get your life kind of straight. You've got some trouble at home, you got a boyfriend that's just a piece of shit, you gotta dump him. Then start taking the classes. You follow what I'm saying? Okay, any questions on that? Okay. And then the other system is the reticular activating system. Now let me kind of describe the terms. This term is an anatomical term. You can actually see this oblong collection of gray matter. When you can see it, that's a reticular formation. It works as reticular activating system. That's the physiology. Reticular formation is the anatomy, reticular activating system is the physiology. It functions in orienting the higher brain at a non-specific arousal state. It could be low or high. But that is done by the reticular activating system. The general level of CNS alertness. General, non-specific. Can I give you an example? Today, this is the way your faces look as I'm busting my ass using all these colors. Lecture exam 1, most of you look like this. Now, if you took lecture exam 1 and you look like this, that might explain your score. You gotta kick your RAS in the ass. You gotta kick your RAS in the ass. So that when you come in on lecture exams, or the TEAS test, or the NCLEX, or the RDH licensing exam, you are sharp as a tack. That's this. Does that make sense? It's game time. Test is game time. Studying is practice. Test is game time. And no one gets pumped for a big game like this.
True.
Following what I'm saying? Okay. All right, that's it for the brain. Any questions for the 5 vesicle brain? Do you see how all that content, even including the integrated systems, were based off the 5 vesicle brain? Busting my ass here, people. Give it back to me. Make my effort pay off on lecture exam 2. I'm begging you to pass this class.
Got it.
All right, now let's go to the PNS. Remember that the CNS is the brain and spinal cord, PNS is everything else. And we have both somatic and autonomic divisions. You guys remember this from anatomy? Somatic, both sensory and motor, are things that you are conscious of. Autonomic, both sensory and motor, are things that you are not conscious of. This is,
Oh my
should be a review of anatomy.
Yes, you did. Okay.
This one I'm putting on the whiteboard is strictly on the motor side. My anatomy students, does this look familiar?
Yes.
All right, somatic side are things that you're conscious of. On the motor side of that, that's only skeletal muscle. That's the only thing you can do on the somatic motor. Somatic sensory are things that you are conscious of, like being touched on the skin. Whereas autonomic motor is going to go to smooth muscle, cardiac muscle, or glands. And there is such a thing as autonomic sensory. You just aren't aware of it. Your brain knows your body pH. Your brain knows your partial pressure of O2. Your brain knows your partial pressure of CO2. Your brain knows your level of blood glucose, your level of blood calcium. Are you aware of that? No, that's autonomic sensory. Everybody got it? Okay. Now, in the motor side, they are all multipolar neurons. So I drew a bunch of dendrites. Kind of looks like an eyelash, huh? those are dendrites. The circle is the cell body, and then the axon and axon terminus. And then I try to tie it in over here. The somatic motor, the first neuron, the upper motor neuron, is in the precentral gyrus. And there's only two neurons here, the upper motor neuron and the lower motor neuron. But if you look on the somatic side, both circles are in the CNS. Is that not what it says here? Everybody see that on the lower left box? But here in this other box, we have to talk about these other terms. There are cell bodies out in the PNS. They are in the ganglion. Remember, ganglion is the word that we use in the PNS. Collection of cell bodies in the PNS. Collection of cell bodies in the CNS should be nucleus, although basal ganglia is an old term that is just probably never going to go away. Should be basal nuclei. And if this is going out, oh, this is going to the heart, guess where this is? The cell body. It's in the brainstem. Vital reflex centers, medulla oblongata. So even when I'm drawing these, elementary sketches, I'm still trying to apply what you are supposed to know for the lectures. Cardiac control center, part of the vital reflex centers, is in the brainstem. It's going to go to a second neuron. We call it the preganglionic neuron, and that's going to synapse with the postganglionic neuron here in green, whose cell body is out in the PNS. That green circle is in the PNS. Everybody see that? This, by the way, people, since I'm a visual learner, this is what I did to my textbooks. My professors didn't have PowerPoints, and they certainly didn't put all this in colors. This is what I did as a student. I got all these words. This is what I did as a student. And I could recreate it, because knowing it is not an option. So here, now you know what UMN and LMN means. Upper motor neuron, lower motor neuron. In the somatic side, there's only the two. On the autonomic side, the lower motor neuron in green, in blue, is called the preganglionic neuron. Do you see why you want to use colors? Look at that, there's all these words. On the autonomic side, the lower motor neuron is called the preganglionic neuron. They're both blue. Does this help at all?
Yeah.
And on the autonomic side, there is a third neuron, postganglionic neuron in green, whose cell body is in the PNS, in the ganglion. That's where the green circle is. And now we can talk about neurotransmitters. Lower motor neurons, whether it is somatic or autonomic, only use acetylcholine. All of them. No exceptions. But on the autonomic side, the postganglionic neuron, it depends. If it is parasympathetic, it is acetylcholine. But if it's sympathetic, and this is an oversimplification, believe me, it is norepinephrine. That's what's coming out of the axon terminals. Any neuron that releases acetylcholine is referred to as cholinergic. Any neuron that releases either norepinephrine or epinephrine are referred to as adrenergic. You know both. Any questions? You see how everything that's in this rudimentary picture is in the last two slides?
Easier to see it.
Again, it doesn't matter whether it's parasympathetic or sympathetic for this guy. Blue ones can only release acetylcholine. Doesn't matter. It's a green one that matters. It can either be acetylcholine or norepinephrine. And this is kind of showing, this picture from your textbook is the bottom half, right here. Everybody see that? Alright, let's just move on. Now, this is now talking about just the difference between sympathetic and parasympathetic. Go ahead and fill in the blanks and let me.
It's just, this is from anatomy. I was just looking through. We have this exact.
Does this look familiar from anatomy?
It's the same one.
It's pretty much the same. There's a couple little additions that I want to highlight.
Neural pattern. Jones.
My goal is to get through, all the way through rod photochemistry today. So you might want to take out the sheet you got last lecture on the essays. That's what I'm shooting for.
It's not freezing.
You guys think that the pace of this class is going pretty quick? Or don't go faster? No? I can go faster?
No,
That's
manageable.
I don't actually have to go this fast in the fall. The reason why I have to go faster in the spring is because of
Holidays.
Martin Luther King Day and President's Day. Do you think that just because you have holidays that you can just cut out shit? It's not optional. That's what you gotta know. You got holidays, that means the rest of the days we're gonna have to move faster. All right, what this is showing you, this is a review of anatomy, fight or flight, or ease and feast. The origin. You should write next to where it says origin, where preganglionic axons come out of the CNS. Second row, origin means where do the preganglionic axons, the blue ones, come out of the CNS? Where does it originate out of the CNS? That's why it's origin. Everybody got that for the second row?
Say it again. One more time.
Origin, second row, means where do the blue preganglionic axons come out of the CNS? Where did they originate? Origin. And you can see here, if this is accurate, what I drew on the whiteboard right there has to be sympathetic, because it's coming out in the middle, auricle lumbar. Parasympathetic is top and bottom. 4 pairs of cranial nerves, 3 sets of sacral nerves. You know what where your sacrum is? You're sitting on it. So it's either top and bottom or in the middle. Everybody got it? Third row. Don't bother memorizing that. just compare it to your own experience of being human. You get spooked, everything dries up. You walk into your mom's house on Thanksgiving, everything waters up.
Yep.
You start smelling good cooking, your pulse rate will actually start dropping. Why? You're going to go through a higher parasympathetic ratio, and it's all shades of gray. People look at this graph and write shades of gray. It's all ratios. It's not black and white, not all one and nothing of the other. It's all ratios. And when you get a higher parasympathetic ratio, like you walk into mom's house with this good cooking, your pulse rate's going to lower, and your mouth's going to start watering, and your stomach might actually start growling. I don't have this row memorized, but I know before a big game, when I'm getting pumped, my mouth is dry, which is good, because I'm going to drink a lot of fluids with maybe some little Gatorade. My heart's doing this right before tip-off. That's fight or flight. Also, fourth row, same thing. When you come into mom's house, it's only your mouth watering and maybe your stomach growling. Your small intestines aren't doing squat. Why? Ain't time for that yet. You start eating, and the duodenum will actually start, just barely get started. Once your stomach is distended, then it kind of gets going. By the time you're squirting stuff out of the pyloric sphincter, now the small intestine's going off. Large intestine still ain't doing shit. Pun intended. Does this make sense? It's going to go in order, because you don't need your large intestines going right when you walk into mom's house. It is selective innervation. Tip off, your whole body's ready to go. Mass activation. For a lot of these things, people, especially when you start having to study for things like T-STES, don't bother memorizing small details. Try to find a way to make this stuff, boom, I can retrieve this. And the best way is to convert it to the experience of being human. And hopefully, you haven't been playing video games all your life. Because if you have, then a lot of this won't make any sense. Bottom row is right here. Any questions? That's, when it says neurotransmitter and effector, the heart is the effector. So this is the neurotransmitter come out, and I would write this down in the bottom row. Neurotransmitters are the postganglionic neurons. Because that's what's going off into effectors from the postganglionic neuron. You can see here, top and bottom, parasympathetic. In the middle, sympathetic. Everybody see that?
Yeah.
Right in the middle. And notice that the organs on both sides of this figure are the same. We call that dual innervation. That's how you're able to have this influence of the shades of gray ratios. Because these organs are getting both. Everybody got it? Your textbook got lazy and just did all the organs once in the middle. So that's how you can see dual innervation. All right. Any questions on this? Now we're going into sensory physiology, and we're not going to complete vision. I want to get through to the essay of rod photochemistry. So does everybody have your essays out? Hopefully I can get to where we end there. Okay. The term refraction means bending light. Refraction means bending light, and there are various structures of your eye that must bend the light to a perfect focus on the fovea centralis. That's way back here. The cornea is the first one, and it bends the light the most. Then behind it is a watery substance called aqueous humor. That bends the light as well. Then it hits the lens, and this is the only refracting media of the four that readily changes. And we're gonna talk about that briefly. Visual accommodation. And then finally, the jelly in the posterior compartment is vitreous humor that also bends the light. So all four of these structures bend the light, hopefully to a perfectly sharp image on the fovea centralis. Everybody got these four? And if you wear contact lenses or glasses, that's because your refracting media is not focusing that image on the fovea centralis. And then you need a fifth refracting media to correct it so that it does. That's your glasses. And when it does this, everyone should know this. On your retina, everything is inverted, just like on this figure. Up is down, right is left. Everything is backwards. And again, what we talked about, I don't know if we have talked about this. Yeah, we have talked about this with intensity coding. When it comes to sensory, and we're talking about the special senses now, your brains see nothing. Your brains hear nothing. Your brain is only receiving action potentials, and if it receives them here, your brain is going to generate a brain-generated illusion of an image. If it comes here, your brain is going to generate a brain-generated illusion of sound. Sound and vision, image, has nothing to do with your brain. It's all action potentials. You guys remember that? Mm-hmm. Well, yeah, so you can actually see it here. Do I look upside down to you? But you can wear these glasses that re-inverts the image so that on your retina, up is up and right is right. You know what that does to you? Reticular activating system. You know what that does to you when you wear those glasses? It F's you up. You have a hard time walking around. You're like, you reach for things over here, but it's actually over here for about 3 days. And your brain will re-invert the image so that up is up and right is right again.
Huh.
Is that wild? That's because your brain sees nothing. It is comparing other senses and reality to the image that it is generating, to the illusion that it's generating, and it's going to reorient it, and finally, up is up, right is right again, until you take the glasses off. And then you're effed up again for about 3 days. Is that wild? Mm-hmm. All right, this is about number 3 on the previous slide here on the lens. Third bullet is about this. This now you must call visual accommodation. Make sure you have that straight. We're not talking about sensory accommodation, which is a decrease in action potential firing rate with constant stimulus. That's sensory accommodation. This is visual accommodation. You have a sphincter muscle, smooth muscle, called the ciliary muscle. It's connected to suspensory ligaments, so on one end of the suspensory ligaments is the ciliary muscle, on the other side is the equator of the lens, right on the equator. Okay, Everybody got that? What I would write next to this slide is this is counterintuitive. This is counterintuitive. So convert it to the experience of being human. This is counterintuitive, so convert it to the experience of being human. Say this right here, biceps brachii. This is intuitive. It contracts, and the tendons and ligaments all get tight. Correct?
Yeah.
This is the opposite when the ciliary muscle, because it is a sphincter. When your sphincter muscle contracts, it closes. So let's hope everybody's downstairs sphincters are right now contracted. When you relax, it opens. Everybody got that? When you contract the sphincter muscle, it closes. So since the suspensory ligaments are here, when this guy contracts, the ligaments get loose. That's the counterintuitive part, right here in these boxes. Biceps brachii contracts, ligaments get tight. And the sphincter muscle, when it contracts, the ligaments get loose, causing the lens to be more spherical, which is its natural shape right here. If you relax the ciliary muscle, all the ligaments get tight and it flattens the lens. See, look at this. Relaxed, suspensory ligaments tight. Contracted, suspensory ligaments loose. Looks of this slide right counterintuitive, because it's a sphincter muscle. Now convert it to the experience of being human. You can watch a baseball game. Those are long-ass games. I was actually more naturally gifted at baseball. But I got bored playing baseball. It's too slow. Hey, I need 4 hours to play a baseball game. And you can watch a baseball game for 4 hours, and it's all distance vision, which means your ciliary muscles relaxed. Your eyes aren't tired. Now study for lecture exam 2 for 4 hours. Oh man, my eyes are tired. Why? You've been studying up here, which means the ciliary muscle's been contracted for 4 hours. Convert it to the experience of being human. You can watch a baseball game, eyes will get tired. Study for 4 hours, your eyes are tired. Any questions? Okay. I'm going to try to get through this as efficiently as possible. When we talk about the rod photochemistry, you're going to have the cells in order. The cells in order in the retina. And the cells of the retina are also counterintuitive. There's a lot in the eye that's counterintuitive. Does this ring a bell from anatomy?
Yeah.
Okay, I'm gonna just plant the seed here for you. If we are talking about things of the human body, anatomy or physiology, and you are bored, you should think about doing something else for a career. If the human body bores you, this is not your field. Now, if we're talking about economic, socioeconomic, governmental policies of healthcare, yeah, you can get bored. That is boring. But if this is the meat and potatoes of how the body works, and it bores you, do something else. Go into banking if you think this is boring.
Look, banking.
The cells are counterintuitively placed. In other words, the photoreceptors are not on the surface of the retina, they are the deepest layer pointed away from the lens. So for the light to hit the photoreceptors, it must first go through the fibers of the optic nerve, ganglion cells, horizontal cells, endocrine cells, bipolar cells, to finally get to the rods and cones that are faced away from the lens. Does this ring a bell from anatomy?
Yes.
So here, be sure you know, like right here, Write the lens on the top of this slide. That's where the light's coming from, in the direction of the red arrow. And the rods and cones are the deepest layer pointed away from the lens. And then the neural signals will go in the opposite direction. It's not action potentials until it gets to the ganglion cell. This is actually an error. But all the neural signals are going up, the light is going down. Everybody see how it's in opposite directions? Here, look at the lights going from left to right. These guys are the deepest layer pointed away from the lens. But then the neural signals are going to go in opposite directions, eventually going through the axons of the optic nerve. All right, now this is the physiology. When we talk about visual transduction, transduction is how receptors convert physical energy into a neural signal. I would write that right now next to the slide. Visual transduction. All rods and cones have a photopigment. A plus B equals photopigment. Chromophore plus protein equals photopigment. The chromophore is a fatty molecule derived from vitamin A. Your body converts it to retinine. Some textbooks call it retinal. Don't get your underwear in a knot over it. For our purposes, it's the same thing. In organic chemistry, they're different, but for physiology, it's the same thing. Okay? Like a fatty acid is a long chain, right? You guys remember glycolysis? I drew out triglyceride. It looks like a really long E. And each one of those horizontal lines is a fatty acid, long chain fatty acid. That's what retinine is. And it's sensitive to light. When it is in the dark, a double bond causes it to bend at the 11 carbon. So it is referred to as 11 cis retinine. It is bent at carbon number 11. That bend makes it fit perfectly on this protein boulder, which is the green. You gotta have chromophore and a protein. Protein is a boulder. In this case, it's a form of opsin, also in green. When light hits it, and here's the transduction, that double bond turns into a single bond and the fatty acid straightens out because it's no longer bent at the 11 carbon. It doesn't totally separate, it just partially separates when light hits it. Everybody see that? That's the dissociation, that's a partial separation. So while it's 11 cis retinine, it hugs the protein boulder. When it's straightened out, it partially separates it. That's the transduction. 11 carbon, see it's bent right there? Now it's straight. You can use, the textbooks also refer to this as the relaxed form of retinine, 11 cis. And this is the excited form of retinine, transol. And use any analogy you want between the relaxed form and the excited form.
Okay.
Whatever analogy works. Now let's talk about the protein. It's one form of opsin, and there are 4 types of opsins. How do I know there are 4 types of opsins? Because we have rods and 3 types of cones.
Oh, right. Okay.
Red, green, and blue. The difference between those photoreceptors is the protein. Okay? Each protein absorbs slightly different wavelengths of light. In the case of rods, the photopigment is rhodopsin. So rhodopsin is the photopigment. We'll talk about this today in lab. A plus B equals the photopigment. And all the chromophores for all of them is the same thing, retinine. In the case of rods, it's retinine plus scotopsin, the protein, equals rhodopsin, the photopigment. Chromophore plus the protein equals. So this is for rods. And it's retinine for all four of them, but you just have different options for the other photopigments. Just fill in these blanks for right now, and we'll come back to it when I get to more visual physiology on Wednesday. First diamond means rods can activate with less light than cones. First diamond means less light needed. Second diamond is explained by the third one. When you have high convergence, we'll talk about that later, it's not a sharp image, less visual acuity. Okay? Okay. Let's just set this up, because I think we're going to have to get to this on Wednesday. You have to have the sequence straight. So if the light's coming in this direction, we're going to start with the rods. And the rods synapse with bipolar neurons. This is the direct sequence. Amacrine cells and horizontal cells are kind of off to the side. So this is the direct inline sequence. You following me?
Mm-hmm.
So this is the rod. It synapses with the bipolar neuron, or bipolar cell, same thing. And it synapses with a ganglion cell. Now what you want to write next to the ganglion cell is that the ganglion cell is a multipolar neuron. Multiple dendrites, can you see that?
Yeah.
One axon hillock, one axon. Multipolar neuron. Everybody see that? Be sure you write that down. Ganglion cell is a multipolar neuron, and it's one axon equals cranial nerve number 2, the optic nerve. And light is actually coming up this way. It's got to go through the fibers of the optic nerve, through ganglion cells, through bipolar cells, to get to the rod and cone, or cone, that's facing away from the lens. So light is coming up this direction. Also, if I drew 3 bipolar cells and 3 rods over here, that's the conversion onto a ganglion cell. Okay? So rods have more conversion on the ganglion cells than cones do. All right? All do this really quick. This will help us go faster on Wednesday. First, you want to write down somewhere that rods and bipolar cells they don't do action potentials. They are graded potentials only. Since there are no all or none action potentials, the only graded potentials, rods and bipolar cells release neurotransmitters based on degree of depolarization. The more depolarized it is, the more neurotransmitters it releases. Rods, and we're talking about rod photochemistry, its neurotransmitter is glutamate. Cones have different neurotransmitters, but all rods use glutamate. And glutamate is inhibitory on bipolar cells. In other words, they hyperpolarize bipolar cells. And again, different bipolar cells have different neurotransmitters. I'm not going to require you to know that.
Whoa.
You have to think this through sequentially. So glutamate from the rods hyperpolarizes the bipolar cell. But the bipolar cell's neurotransmitters depolarize the ganglion cell, and the ganglion cell, if it reaches threshold at the axon hillock, will fire the action potential. Where? Down the optic nerve. Because that's what its axon is the optic nerve. So that basically covers this slide here. Rods and cones and bipolar cells with graded potentials only. They release neurotransmitters based on degree of depolarization. Okay? And so when we start on Wednesday, you need to take a picture of this. This needs to be up when I start on Wednesday, because you need that to understand the essays on rod photochemistry.
Okay.